Okay, good afternoon, everybody. Thanks for introduction from Edmund. Uh, Thanks to be here in the beautiful city. Thanks to ProImmune organizers for giving me an opportunity to be talking to you all on our own industry experience, what we have gone through in the last, say, 5 to 6 years. So I'm from Dr. Reddy's Laboratories, India, where I have been part of this company for last 24 years or so. Started my career as a veterinary pathologist and then got into the new drug discovery and then got into the differentiated product development program where we were focused mainly on the 505(b)(2) applications. And then in the last 4 to 5 years, I have been part of the generic drug development efforts, where I am extensively involved in the toxicology support to various product development. So when Edmund approached me to give a talk here in this conference, I was wondering what is that I'm supposed to be doing here. One of the things is that, I knew that ProImmune themselves are, super experts in carrying out the assays, conducting the assays, assay interpretations, and they would be definitely talking about their assays, which we saw in Edmund's presentation in the morning. Then I thought, what is that I have to give it to you people? So what I'm going to speak, this is a very general presentation, basically focused mainly on, what is our experience from an industry perspective, what exactly we have learned in last 4 to 5 years. It is little more than, 5 years we have been working in the generic peptide drug development area. I think we have evolved ourselves quite significantly, I can tell you, in the last 5 to 6 years, thanks to various agencies. We have been dealing with FDA, EMEA, and also Canadian agency. It's been a significant learning experience. Of course, it's been pretty challenging, and we have seen some success. But I'm very sure the way we are evolving, where we are progressing, I'm sure we will have definitely much more successes as we move forward in the next few years. So that with brief introduction. So I will be moving to how am I going to cover this presentation. This is basically many of these contents, whatever you see here, are partly being covered by speakers. Edmund covered something, Tim covered quite a bit in the morning, and like that. This session, what I will be talking to you more on the, why is this immunogenicity matters in the generic peptides, and what are the things that drives the risks, and how to assess this risk, and once you make the assessment of this risk, how do you really manage and mitigate this risk, and real world pitfalls. What I mean to say here is I have some very good examples of our own learnings, which of course some of them were bitter learnings, which we realized at a stage where it should have been picked up at the early stage, and finally, what conclusions we can derive out of our experience. So, when you have the peptide product development, drug development program in your portfolio, as everybody follows certain guidances. Here in the peptide area also, we follow the guidance that is from the FDA and EMA. I'm sure you must have gone through these guidelines. Very important. What do they expect normally? To very briefly to tell you, their expectation is mainly as far as toxicologist, as for me is concerned, the safety and of course the efficacy part. So where is this safety issues coming for you? It is definitely the impurities arising over, be it the process, during the process or the peptide synthesis at various stages, correct? So once, the expectation from the agency is that you identify those impurities which are not, whether it is a new impurity or is it an impurity which is already present in your reference drug, or is it to what level it is present? Is it, if it is present anything above 0.1%, you will have to deal with it differently. So once you identify those impurities, the in silico evaluation will be performed. What is in silico? You know that you have to understand the impurities potential to bind, its potential to bind to MHC binding T cell epitopes, which again, will trigger the immunogenicity, immunogenic response, which I will be talking a little more in detail subsequently. So everything, so these are the expectations from the agency. And one size doesn't fit all. So you need to address this with case by case basis. So, there are certain, every time you cannot say that I will do this assay, and I will close it. You need to really look into the multiple approaches to, address this risk. So then you develop a strategy, identify those risks and mitigate those immunogenicity risk to prevent any late stage surprises, which we ourselves have gone through. That is where I am telling you, you need to be very careful from the beginning of the development itself. Then one thing I just want to make a comment here, even though you have the guideline here from agencies, but there are no harmonized guidelines like an ICH guideline or EMEA guideline, where I'm going to just flash you one slide. What is that from the industry? What we need, from the regulators. So for toxicology, if you ask me, it's more than 40 years we have evolved significantly. You have an ICH guideline, Q3A, Q3B to impurities, the moment it reaches beyond whatever is prescribed in the ICH limits, what to do, correct? You do the in silico analysis, you do a number of assays to be performed for genetic toxicology, qualification of the impurities. So, very set process is available. But of course, we have to understand the fact that toxicology evolved over more than 40 to 50 years. But I'm sure we will get there even for the peptide impurities. So moving on, then this is the expectation from the agency. Then what drives the risks for us, for the companies who are, involved in the product development? So the first and foremost thing, what we need to understand is that there are, various classes of peptides. It could be cyclic peptide, linear peptides with lipophilic chain, and the random polypeptides and the pegylated peptides. So just to give a very snapshot of what is the small differences exist between them. The cyclic peptides have got a very low immunogenic potential propensity due to their rigidity and the fewer exposed epitopes. So for example, some of the examples here is the calcitonin, vasopressin, octreol, landrio, and telepressin. Here I can proudly say that these two products, we already have the approval. As I said, there are we did not really face the challenges what we are facing down below the list what I am presenting here. The linear peptides, and again these linear peptides also have the low intrinsic immunogenicity. They don't create so much of a problem, but definitely sensitive to impurities and the sequence changes. So some of the examples are these two, and also the teriparatide. This is another product which, of course, we are working. As I said, this is the product, our own experience. We have been able to address the immunogenicity challenge, but unfortunately, we do have challenges in some other areas of teriparatide. So this is an interesting, peptides for all of us. I think the entire universe is around these linear peptides with lipophilic side chains. These are the GLP-1 analogs. I'm sure, you are all very well aware of the blockbuster drug, semaglutide, and the liraglutides, and, tirzepatide, retatrutide. I mean, these class of drug definitely they do enhance the immunogenic activation, but to a certain extent, definitely it can be controlled. That's what we have learned. But I can tell you, we still haven't surpassed the challenges, immunogenicity challenges, these drugs, what we are facing as of today. Then we have random polypeptides. These are very highly variable with multiple epitopes, and induces the enhanced immunogenic responses. The best example is glatiramer acetate. Yes, we do work with this glati, but here we did not see any immunogenicity concerns. Of course, there are other challenges. Similarly, I think Tim mentioned today morning, pegylated peptides. These pegylation, whatever the additional things you are making with the peptides, mask the peptide epitopes, but you are likely to end up with the introducing the anti-peg immunogenicity risk. So now, moving on, just remind me of the time, please. What are the sources of the impurities across the generic life cycle? So when you looked at the guideline, you looked at what are the different classes of peptides, and then what are these sources of impurities during the life cycle of your peptide drug. So this the source of impurity could be coming from the raw materials what you are using, and during the synthesis process or during the purification. The scale dependent. This is very important. At the R&D stage, whenever you are working in the lab, you may not really see the impurities rising up. The moment you get into the scale up at the plant, you will suddenly see surge in the impurity. The levels are going up. And further, API, there are API and the formulation. The API is active pharmaceutical ingredient. So, the stability of the API itself, further enhance the level of the impurities over a period of time. So similarly, the degradation, during the stability. Similarly, when you move, this is basically, as I said, these are all the drug substance related. When you move into the drug product, you are, definitely at the formulation stage. When you have made the product, and during the stability also you can encounter it could be a new impurity, or it could be an existing impurity similar to that of the RLD may start growing up. So these and some of the other sources includes the modifications. The moment you introduce the modifications, the addition, deletion, truncation, all these modification has the potential to aggregation. All these can, introduce the immunogenic response. So it is not just the API and the product. So there are many other factors which are playing a very critical role in generating the impurities. So moving on, then you once you understand where are the sources of impurities coming up. So you need to understand what are those different factors influencing the immunogenicity. So it could be product related, or it could be treatment related, or it could be patient related. Tim did flash a slide where it contained most of this information, but I'm going a little bit, I will be talking on this, which he just flashed. Thanks for that. So you can see this. This is more focused on the product related. The peptide synthesis and the route. What is that route of synthesis you are following? It could be solid phase protein peptide synthesis, or is it a recombinant process, or are you following the hybrid approach? So whatever is the synthesis route is very critical for you, and this is if it is not taken care in the beginning, you may end up, to start with, you may start having these impurities in this stage itself. And this could be a same process of the as your reference drug. Same again, if that process is different, so you will end up in the issue. Then again, the sequence modification. You tend to have you see this in this era of peptides. uh, the landscape is so high that you need to do all those things to get to it. There is a very subtle difference between you and the innovator for the generic drug companies. So some small modifications you introduce, it could be natural versus the non-natural amino acids, leading to epitope alterations and then leading to the impurity formation. Then again, structural and conformational changes, it could be tertiary or secondary changes leading to impurity. Similarly, solubility and stability is also very important. When you want to look at the impurity, solubility of the impurities for your assays, it is very critical that your impurities are soluble in the media, not impacting the media, leading to the sensitivity of the assay being impacted. Similarly, the sterility and contamination. I have one big learning from here for us, which I will be talking about, these endotoxins and the microbial risk. So these are all about the product. Similarly, analytical gaps and challenges. This is another area. Whenever you do the adopt the analytical methods, you will end up in challenges when the different peaks are coming up during the analysis. So treatment related. So what is the treatment related? Again, different routes of administration of your drug, frequency of administration of your drug, and it could be IV, it could be intravenous, intramuscular, subcutaneous, or oral. Just to make a mention here, so far there are no immunogenicity requirement has come so far for the oral drug, because just to tell you simply, which is when compared to the intravenous or the parenteral route of administration, orally the peptides gets disintegrated, and so far they have not posed the challenge, so the requirement has not come up. And then the last one is the patient related. I think some mentions have already been made. What is the patient status? What is the disease condition? What are those MHC alleles which they are having? And what is the immune status of the patient? All these factors play a very important role in the immunogenicity potential. Then, so as I said, even a very minor change in the synthesis of the structure or the formulation can significantly impact the immune response. So moving on, how do you assess this risk? So integrated approaches, when we talk about the non-clinical immunogenicity, already, we did touch upon the in silico and in vitro assessments. What do we do at Dr. Reddy's is that we start with the in silico assessment, and if you have identified, if the low immunogenicity risk is identified, we will do some risk-based justification. We do not get into any other no further studies. But even though you do the in silico analysis, it just doesn't end here. Even though you prove that you have the low immunogenic potential, you must have to get those impurities through in vitro assessment. This is a mandatory requirement, and which is the approach we are also still abiding at Dr. Reddy's. So again, at this stage, when you do the in vitro assessment, if any high immunogenicity risk is identified, we definitely take them through the risk-based justification, and wherever required, if additional studies may be required, or if it is if the potential is high, you may have to go back to the in silico evaluation and see whether either it can be controlled, or if you are not able to control, and whether you want to re-look into the entire processing. So then, little bit more on the in silico and the in vitro assessments. I will be. Why do we do this? This will help us in predicting, and then once you predict it, we rank order them and then prioritize them. So how do we take this? So before we even get into the entire cascade of in silico analysis, there is a group which is continuously working on the API characterization in our company, where always the focus is on what is the impurity levels, always compared against the reference drug, whether it is the new impurity or whether it is present at the above the level of the reference drug. And you just start playing here itself. You can really go back to the bench and see whether certain modifications can be applied. It is because nowadays the companies are offering the in silico along with the ML modeling, AI modeling. There are a number of options available for us, so we put quite a lot of efforts in this stage itself. Then what happens? So once what happens in the in silico, basically you retrieve all the impurity sequences, then you scan it through then you look for what is its binding affinity towards the HLA alleles. So it should be, the alleles should be of representation of different population. Whereas once you identify the binding affinity, then you get into the screening of the T cell epitopes. Just to what happens within the in vitro system is, once these peptide gets recognized by the antigen presenting cell, you will see the antigen presentation happening, which what you are seeing here is the binding pockets, where these impurities are coming and sitting over there in the binding pockets, and then attracts the T cell population, T cells, and then further immunogenic cascade gets triggered. So then once this is what happens within the system. Whereas in the in silico also we try to mimic this, where once you have identified this T cell epitope, you compare this with the universal database which is already available with the tools, and then you try to understand the humanness. That means how close these epitopes are to the humans. You compare it and try to rank order this. So whether this T cell epitope it is present, if it is more humanness, then it is more likely to induce the immunogenic response, and it is if it is present, but if it is less human, it is less likely to induce the immunogenicity. Like that, you try to rank order the the impurities which you are taking it through this analysis. So finally, it is very important, this entire exercise, which will tell you a fair idea of immunogenic or the non-immunogenic risk assessment at the beginning stage itself. So, as I said, even though you do the in silico, you will have to take it through the in vitro assay. So, I think, Edmund spoke quite a lot on what are the different assays they have, what are the principles of those assays. It is something which I don't think I would like to, repeat the same. These are something, once the impurities get exposed, it will be extracellular, it could be attracted by, processed by the extracellular or intracellular sensors, in turn activating the nucleus and then CD4 cells, and then followed by, naive CD4 cell activation. See, immunogenicity cascade within the body is multifactorial. You cannot really, you won't be really predict what is the cascade it is going through for a particular drug. So this is a fair understanding which we have. So, let me skip this because he has already spoken. Finally, what I want to say is anti-drug antibody production happening, which can be really, evaluated even in the clinics also. Then, again, you know that innate immunoassays and the adaptive immunoassays. Innate PBMC stimulation assay, which we work with ProImmune extensively in the last many years. They have been our, collaborators for this assay. And how to manage and mitigate, as I think this is slightly repetition. I have another 5 minutes just to share you a few of our own, examples. I would skip this because, as I said, what I tried to tell here is It is from the development phase itself, we try to eliminate or control the impurities. We just don't get into this filing phase immediately, because this has come from over a period of experience. We try to get the handle on the impurities at this stage, then followed by the confirmatory phase. Even at the confirmatory phase, we try to do some exploratory studies to get a confidence on the impurities not having the immunogenicity potential. Then at the product filing phase, we will be entering into the full-fledged, in vitro assays, whether it is innate or the adaptive immunogenicity assays. Now, coming to some of these examples, I would like to share this with you all. This is our own learning. This is not coming from any publication or anything. So high dose of impurity testing than the realistic concentrations. So this is one of the critical expectation from the agency also. We used to, 2-3 years ago, we used to do the adaptive assays using the product. Then we gave a very clear response from the agency. We got that, you should not be using the product, but you should be using the individual impurities. So when you use the individual impurities, you must have a handle on what is that concentration you are going to use in the assay. It should, see, the level of impurity present in your product could be less than 2%. But when you use it in the assay, the concentration what you are going to use is way above what it is going to be present either in the product or the amount which is going into the humans, in the human body. So, what it does, what is the pitfall problem of these high dose impurities? If you don't have a control, it leads to unnecessary immunogenicity signals. We have faced it. It introduces the non-specific binding and the assay interference, and then it leads to exaggerative adaptive immune responses. And finally, this is very important for you. Then worst case scenario, we have ended up in a situation where we saw the immunogenic potential without realizing that it is because of the high dose, high concentrations we have used. We have gone back to the drawing board, try to control them. Imagine, if you in a product development, if you want to really get into controlling the impurities, it is just not the time, but it is lots of dollars. So this is our own experience, and we have learned and we have now got a good understanding of how this should be defined within the assays what we are carrying out. Then moving on, endotoxin. This is very important. This is our own learning again. When the product is being developed, the product development people, they have certain specification. This is all the USP specification defined based on the route of administration and the threshold of the pyrogenic risk. So when they do it at the plant, these are the limits, they think about. They are not really worried about, they were not really aware of this impact on our immunogenicity assays. So in the immunogenicity, it should be less than 0.01%. So the moment, less than 0.1 EU per mg. The moment your level goes beyond this, you will end up in unwanted immunogenic response. So we realized this, we did. It took us almost 6 to 8 months for us to resolve this issue, because when the impurity is being synthesized, you don't really care. They were not, I don't say they don't care, they were not aware that this is a critical part. So we took some time to understand this, and again, when you started individual impurity synthesis under a controlled environment, when we resolved this, and we knew that the impurity is not immunogenic. So imagine the time and the money by then we had spent. So the learning came with significant cost and time for us. Sorry, another 2 minutes? Yes. So this is another real-time example on the solubility. So when you are testing the impurity in the adaptive assays, the solubility is very, very important. Sometimes you may have to use the component which are not part of your formulation. Say for example 1% ammonia. The moment here the agency expectation for us is that you should not be diluting the impurities too much. It should be tested at a certain ideal diluted concentration. So this is not always, in certain cases it happens. The impurity at the higher concentration will start showing the precipitation. So this will lead to false immunogenicity signals. So you need to be, this again, this is our own experience. So we need to be very careful in choosing the component of the impurity, the solubilization. So this is also another important thing. Then moving on, this is the last one, the excipients. What you use, these excipients, can blunt the responses. There is a, paper published by the FDA where very clearly state that, masking the presence of immunogenic innate immune response, this is the IRMIs, thereby reducing the assay sensitivity. So this is again, which we have experienced. If you see the phenol, if you look at this assay, the positive control, which doesn't have the phenol, it has given 75% response, whereas in the blank you have seen only 2% response. But with the product which contained the phenol, it shows 2% response, which is equivalent to the blank. So clearly indicating that your excipient phenol is masking the immunogenicity response. So the moment when we removed the phenol from the product, you see 15% response. So you need to be very, very careful about choosing the excipients for your assay, which is compatible with the in vitro testing systems. So moving on, I think I'm coming to the last slide. So just to conclude, risk is definitely multifactorial. It can come from anywhere. It could be structure, impurities, formulations, assay conditions. It is just not single factor. So as I said, integration is very essential from the beginning. It is not just in silico, not just in vitro. You need to be spacing the strategy, at every step. Then assay design dictates the outcomes. As I said, the dose, endotoxin level, solubility of the excipients leads to false positive or false negative immunogenicity conclusions. Lastly, immunogenicity assessment beyond borders. Now, I think I did mention about this global harmonization of the guidelines. This is very, very important. So I just don't want to go through this. I'm already completed my time. The final thought is that the goal is not just the data generation, but interpretable immunogenicity insights that enable safety-focused decisions for the generic drugs. Thank you very much for patient listening. I hope I'm...
I have a question. Do you have any differences in impurities from batch to batch?
Pardon?
Do you have differences in impurities and impurities quantities and etc. from batch to batch of generation of your peptides?
Very good question. Batch to batch variation of the impurities you will encounter if you are synthesizing the impurities in your lab. We have come across this, but we haven't seen so much of a differences between the batch to batch. And also, one I think insight I can share you is that when we realized this endotoxin issue, when we had sourced it from the outside source, we did not really had this issue of the endotoxin. But when we were doing it in-house, yes, this was there. I mean, I just little deviated, but if your processes are controlled, you are not likely to end up in the differences.
Okay.
So much.
Thank you.